Assalamu alaikum once again. I'm back. Please remind me of the fact that when was the last recording posted? Cell membrane का part 2 कब post हुआ था? Because in that I have covered the entire chapter. Okay, so मुझे एक दफा बता दे, because if you have not listened to the recording, then you'll be unable to answer these questions. तो मुझे उस्मान अब्दुल रहमान थोड़ा सा बताएं कि आप लोगों ने रिकॉर्डिंग सुनी है नहीं सुनी है.
मिस मैंने स्टार्ट की थी और हाफ हो भी गई है। बस जो मूवमेंट वाला पार्ट था ना वो
All right, so you want me to give a recap of the chapter or you want me to start the past paper right now?
मिस वी कैन स्टार्ट पास्ट पेपर्स।
ठीक है, और अब्दुल रहमान आप कह रहे हैं कि थोड़ा सा रिवीजन कर लूं.
यस मिस। कैरियर
मुझे
प्रोटीन तो जो इंटेग्रल प्रोटीन है ना, उस साइड से मैंने देखा था कि
अच्छा, मुझे ये बताएं कि आपको किधर से क्या चाहिए, ताकि हम वहां से करें और फिर उसके बाद पास्ट पेपर्स करेंगे, बिकॉज उसके बगैर तो हो ही नहीं पाएगा. सेल सिग्नलिंग
नहीं मिस, इससे पहले जो प्रोटीन्स वाला था ना, कैरियर प्रोटीन्स वाली स्लाइड था मैं।
साइड पे थे.
हम्म,
ये प्रोटीन तो बहुत ऊपर था.
हाफ वीडियो होगा जी उसमें, और उसके बाद शायद ये, हाँ ये वाली स्लाइड था भाई। ये बिल्कुल लास्ट स्लाइड पार्ट से था मैं, और उसके बाद उसकी खत्म होने वाली थी स्लाइड, तो आके बस वहाँ रुका हुआ है अभी।
अच्छा चलें, ठीक है, इसको कर लेते हैं, बट चलें फिर ब्रेक से पहले हम पूरा रिवीजन करते हैं एंड देन ब्रेक के बाद विल डू द पास्ट पेपर्स. ठीक है. अच्छा जी,
जी।
सो हमने जो है वो ये देखा था कि इस तरह से समझा रही हूँ that we have phospholipid bilayer. Phospholipid bilayer क्या होती है? It's cell surface membrane or plasma membrane. We will not use the word cell membrane up say now in A levels up till your A2s. ठीक है? Use the word plasma membrane, phospholipid bilayer or cell surface membrane. And phospholipid bilayer, if we break this word into two, phospholipid. So phosphor means phosphate ka head. So this structure, the circular structure is phosphate ka head, right? And then this is phosphor. The lipid part is the fatty acid tails. Two fatty acid tails are projecting out from this circular region, and these are fatty acid tails. Okay? Then we talked about the fact that your phosphate head is hydrophilic in nature, and if we break this word hydrophilic in two, then hydro means water and philic means water loving. Philic literally means loving. So this is why we call it water loving. The phosphate ka head is water loving, while these projections that are coming out of this phosphate head, these are hydrophobic in nature, and hydro means again water, while phobic means fearing. Okay? So this is water fearing. These projections, these fatty acid tail projections are water fearing, while the phosphate head is water loving, hydrophilic. Okay? And I also mentioned that fluid mosaic a question, it will come into your paper for sure, 4 marks. So this is the entire detail that I'll tell you right now, you'll have to write in your script. Okay? Do not miss out on any of the points, because then you'll lose your marks and then you'll say, okay, great, Aya, because I do tell the important keywords that should be a part of your script side by side. Okay? So it's starting off with the structure of the plasma membrane, and this question will be like, describe the fluid mosaic model of phospholipid bilayers. The fluid mosaic model is that a plasma membrane has a phosphate head, and with A2 projections of fatty acid tails are projecting outwards, and they are hydrophobic in nature, while the phosphate head is water loving or hydrophilic in nature. Then we have certain molecules. In these molecules, these big molecules are protein in nature. So we have basically two types of protein in phospholipid bilayer, integral proteins and peripheral proteins. So integral proteins are of two types. They are channel protein and they are carrier protein. I'm just quickly going over through the entire concept because I've already covered all of this in the previous recording. But as a part of revision, we'll do it again. Okay? So integral proteins are of two types, channel protein and carrier protein. Channel protein basically has a channel in between. This is a channel. Okay? While a carrier protein I said that when I made this diagram in the recording, I said that carrier protein somehow looks like this. Okay. And when a molecule, it has to go inside the cell, then it binds with the carrier protein, which induces a conformational change within the carrier protein, and then it will move towards the inside of the cell like this, and the molecule will move inside of the cell. Okay. This is how your carrier protein works. But this is not how your channel protein will work. Your channel protein is something like this, and it provides a pathway for ions or molecules. So this is the pathway for ions and molecules provided by the channel protein. Okay, so with channel, try to remember this word, that channel is providing a pathway for ions and molecules to project inside the cell. And channel protein, carrier protein, they're both the components of integral proteins, meaning they're inside the membrane, while peripheral proteins are outside the membrane. They are towards the surface of the phospholipid bilayer. Okay. Next, we have a cholesterol molecule, and it plays an important role in fluidity, in maintaining the fluidity and stability of the membrane. And then we have certain projections that are moving outside the cell surface membrane. These are made up of two components, carbohydrate and protein. These are known as glycoprotein or glycolipids. Okay. So glycoprotein means that glyco region is carbohydrate wala part and protein is protein wala part. Similar projections, we do have similar projections somewhere else, and they are known as glycolipids. Okay. So we'll cover the function of each one of them. This is just the brief introduction of the structure of the plasma membrane. Okay. A quick check that I told you the size of plasma membrane. What is the size of plasma membrane?
7 nanometer.
7
7
nanometers.
nanometers. Excellent. 7 nanometers. Okay. And now, so far, what we have discussed the structure of the plasma membrane, in which we have phospholipid, and in that we have phosphate head, we have fatty acid tails. We talked about the nature of both of them. We talked about two types of protein present in phospholipid, that are integral protein and peripheral protein. Integral proteins are inside the membrane, while peripheral proteins are outside the membrane. Then we have two categories of integral protein, channel and carrier. Channel protein basically provides a pathway, while carrier proteins bind a molecule within itself and then induces a conformational change. That molecule induces a conformational change within carrier protein, and then it turns upside down and enables the molecule to move inside the cell. Then we have cholesterol molecules, which play a role in maintaining fluidity and stability of the membrane. And then we have certain projections coming out of the cell surface membrane, which are glycoprotein and glycolipids. So far, are we clear?
Yes, miss.
All right, perfect. Then I call this plasma membrane, the second name of plasma membrane is phospholipid bilayer. So why are we calling it phospholipid bilayer? It is because of the fact that there are two layers of this membrane. So this is the first one and this is the second one. So these projections, these fatty acid tail projections, they are also forming a hydrophobic core. Okay. So phospholipid bilayer means that they are arranged in a bilayer manner. They are arranged in two layers, and the fatty acid tails of two layers, they combine and they form hydrophobic core. Okay. So far, are we clear? Then we have the fluid mosaic model. Okay. So fluid mosaic model literally means that your phospholipid or your cell surface membrane is not static. It's not in a solid manner. It is in a fluid manner. Okay. So it has some fluidity, because cell surface membrane cannot afford to be static. It cannot afford to be in a frozen manner. Rather, it has to be fluid. So the molecules and the ions can move inside the cell easily. Because if something is solid, then it's hard. It is harder for ions or molecules to propagate inside. While if something is in a liquidy, in a fluidity manner, then ions and molecules find it easy to propagate inside. Okay? This is exactly why phospholipid bilayer is in a fluid manner. Then you'll be asked to describe this entire word, fluid mosaic model. It's written over here. So fluid mosaic model refers to the movement of phospholipid So this happens because phospholipid is in a fluid manner, it is in a liquidy manner. Then the word mosaic literally means that it has a scattered proteins over the entire surface of phospholipid bilayer, meaning that it has integral proteins and it also has peripheral proteins. So they are in a scattered manner. They are not in a fixed manner, they are in a scattered manner. And this is why it is giving an appearance of a mosaic. Okay, so fluid mosaic model means that fluidity is influenced due to the factor of phospholipids, while your mosaic is due to the scattered proteins all over the surface. Okay, we already talked about this. We have talked about this as well. I mentioned that we have certain projections, certain chain-like projections that are coming out of the cell surface membrane. They are glycolipids and they're glycoproteins. We talked about the intrinsic protein. Now, moving towards the categories of intrinsic protein, we talked about channel proteins, we talked about carrier proteins. Receptor proteins are also intrinsic proteins, and they play an important role when a cell has to send a signal inside. So the entire process is known as cell signaling. Okay, cell signaling basically a signal comes to the cell and it has certain message. Then it binds with its receptor protein, and then that receptor protein enables the signal to move inside the cell and then cause whatever change, whatever metabolic change it has to cause. It is caused due to cell signaling. Okay, we do have a separate subtopic of cell signaling, so this is just a brief introduction of what receptor protein and what cell signaling is. Generally, try to remember this word ligands. It's an important word, and you have to mention it in your script as well, that ligands are all those molecules or all those signaling molecules that bind to the receptor protein. Okay? So they are ligands. They could be any neurotransmitter, they could be a hormone, they could be an enzyme, anything which binds to the receptor protein and cause any metabolic change within the cell is known as ligands. Okay? Then we have peripheral proteins. I mentioned that they are not embedded. They are found on the surface of the cell surface membrane. And this is it about peripheral proteins. Then we have cell signaling. So cell signaling is an important topic. It usually comes from 4 marks. We'll do it in the past papers as well. For cell signaling, your peripheral proteins are involved. Okay? And what are they involved in? They're involved in transducing the signal within the cellular components. Okay? And peripheral proteins are also involved in enzymatic activity. So remember, these are the functions of peripheral proteins. That peripheral proteins are involved in cell signaling. They're also involved in enzymatic activities. So basically what they're trying to say is that peripheral proteins also act like as an enzyme. So they are able to catalyze reactions that are happening on the surface of the membrane. Okay, now this is the important part as well, that factors affecting fluidity of membrane. So I mentioned that membrane is fluid in nature. It's not static, it's not in a frozen manner, it is fluid in nature. So fluidity of the membrane is influenced by tail length, saturation of fatty acid tail, and because of temperature. So there are 3 factors that influence the fluidity of the membrane. Number 1, tail length. We said that phosphate has tails, right? Phosphate has tails. But they are saying the longer the tail, the lower the fluidity of the membrane will be. So you need to remember the fact that the shorter the tails, the more will be the fluidity. Okay? So whenever you'll have a long tail of fatty acid, then your fluidity of membrane will decrease. But when you have shorter tails, the fluidity of membrane will increase. Then I mentioned saturation and unsaturation concept in the recording, but since you have not listened to it, let's talk about it again. So in chemistry, back in your O levels, you studied single bonds, and you studied about double bonds, right? पढ़ा था कि नहीं पढ़ा था?
पढ़ा था।
इसमें से अच्छा तो पढ़ा था, तो बताएं which one is saturated and which one is unsaturated.
हमें single bond saturated है और double bond unsaturated.
Exactly. Double bond is unsaturated. Why? Because if we have, for example, bromine ion. Bromine is Br2, right? Yeah, chlorine ion Cl2. So bromine or chlorine, they have the ability to break this double bond, or both double bond break karke, they'll bind with carbon and carbon. Okay? So unsaturated means that this molecule can be broken, यानी इसका bond can be broken and other molecules can attach with it. While carbon-carbon का single bond is showing that this molecule is in a saturated manner and no other molecule can come and bind with this. ठीक है? So unsaturation basically is trying to tell us that more molecules can bind with it after breaking the double bond, while a single bond cannot be broken further since it cannot be broken further and no other molecule can bind with it. So this is saturated. Okay. So a higher degree of unsaturation in the tail leads to increased membrane fluidity. Okay. So remember, this is a very important part. कि जितना your tail is going to be unsaturated, it is going to increase the membrane fluidity. Okay. And इसका थोड़ा सा technical aspect, if I try to teach you, I did not mention this in recording, मुझे बस अभी याद आया कि ये देखें, if the projections of fatty acid tails are like this and if the projections of fatty acid tails are like this, bendy है, ये straight and this is bendy, which one do you think is going to influence the fluidity of the membrane? Bendy वाला या straight वाला? फ्लूइडिटी मेम्ब्रेन किसमें ज्यादा होगी? See, when you don't answer, I feel that I'm teaching myself. Let's boiling.
Unsaturated में.
Unsaturated में. तो unsaturated कौन सा लग रहा है इसमें?
Double bond.
Yeah, this is unsaturated. जितना bendy होगा उतना unsaturated होगा. जितना saturated होगा उतना straight होगा. तो straight वाले में इसमें can you see कि इतने straight है, इसमें fluidity होगी ही नहीं. Bendy होगा उसका structure, तो उसमें fluid का influence ज्यादा हो सकता है, उसको move करवा सकता है, right? So this is unsaturated. This is saturated. तो ये unsaturated है, bendy होती है unsaturated fatty acid tails. So the more bendier they are, the more the more fluidity can they achieve. Okay. So the higher the degree of unsaturation or higher the degree of bendiness within the fatty acid tails, your membrane fluidity will increase simultaneously. Okay. And at low temperatures, this is also very important because this may come not just as a factor, but this may come as a role of cholesterol as well for 2 marks. Okay. I've seen this question repeatedly coming in your papers as well. When you'll do it, you'll also understand कि कौन से questions जो हैं वो ज़्यादा important. So this may come as the function that state the function of cholesterol for 2 marks. ठीक है? So try to remember this. That at low temperatures, cholesterol will increase the fluidity of the membrane, preventing it from being too rigid. Like I said, that cell membrane cannot afford to be rigid or it cannot afford to be in a frozen manner. Rather, it has to be fluid in nature so that molecules can easily propagate inside the phospholipid bilayer. Okay? And at high temperatures, cholesterol will decrease the fluidity of membrane and will stabilize the cell. So cholesterol is basically playing 2 roles. It's playing a role in maintaining the fluidity and it's also playing a role in maintaining the stability of the cell. Okay? So small components of cholesterol can be seen within the phospholipid bilayer. They're attached at different instances and they are helping the cell to achieve stability and fluidity. Okay? So low temperature पे याद रखना है that at low temperature, cholesterol will increase the fluidity of the membrane. Why is that so? To prevent cell surface membrane from become too rigid. ठीक है?